Welcome to our very first video. If you're excited to explore the fascinating world of biochemistry with us, make sure to hit that like button and subscribe to the channel so you don't miss any of our upcoming content. Let's dive in.
Another exciting episode of Biochemistry Simplified. Today we'll be looking at glycolysis overview. This is really extremely exciting as we are going to be starting a new topic today. We are going to start with another aspect of biochemistry, which is metabolism. This is the section that we actually test your memory. There's going to be a lot of reactions going on. There's going to be a lot of enzymes, a lot of mechanisms, but all of it is going to be absolutely beautiful, trust me. Now we move into glycolysis proper. In this lesson, it's just going to be an overview of the whole process of glycolysis. I'm going to be talking about the glycolysis in general, and subsequently we'll go through the details, talking about the individual steps of glycolysis, the structures, etc. Now we'll begin with glucose. Glucose, as we know, is the primary sugar monomer that your body uses to create energy. That is what is ultimately metabolized. That is what is broken down and run through the metabolic pathways, and all of the energy is extracted from glucose. It is used to make the adenosine triphosphate, which is your ATP unit. All of the carbons are oxidized to carbon dioxide, CO2, and it's subsequently oxidized to water. But it all begins from with glucose. Let's take a look at what glucose can do. As we can see, glucose has three primary parts that it can follow. When we say primary, most of the glucose goes through three pathways. As you can see, um, they are they go through many other pathways, but these are the major three pathways that glucose can go through. One of them is the storage pathway. As you can see, glucose can go through the storage pathway, whereby it is stored to starch and glycogen, and that's simply for storage purposes. Glucose can also go through the glycolysis pathway that we are currently examining, and the end product of that pathway is pyruvates and what energy in form of ATP. And glucose can also go through the pentose phosphate pathway, whereby we have ribose 5 phosphate and NADH as the end product. Recall that NADH is the reduced form of the coenzyme NAD plus. In glycolysis, a molecule of glucose is broken down in 2 molecules of pyruvate in 10 enzyme catalyzed steps. Of course, we know that every reaction of the body is catalyzed by an enzyme. So for glycolytic pathway, we have 10 enzymes running through that pathway. Glycolysis can also be considered in majorly 2 phases. In glycolysis, a molecule of glucose is broken down into 2 molecules of pyruvate in 10 enzymes catalyzed steps. Yes, 10 enzymes catalyzed steps. Now, those 10 enzymes catalyzed steps are classified or considered in glycolysis under 2 phases. We have the preparatory phase and what? The payoff phase. The preparatory phase, a molecule of glucose undergoes through 5 enzymatic reactive steps to produce 2 molecules of glyceraldehyde 3 phosphate. And in the second step, known as the payoff phase, the payoff phase, those 2 molecules of glyceraldehyde 3 phosphate undergoes another 5 steps to produce 2 molecules of pyruvate. That is how glycolysis is broken down to 2 in 10 enzymes catalyzed steps. Now, the preparatory phase is also known as the investment phase, whereby two molecules of ATP, that's energy, is invested in the process to produce four molecules of uh, ATP in the payoff phase. So you see a little bit of symmetry that the 10 steps has been divided into. Again, we talk about two phases: the preparatory phase, the payoff phase. Please note that these are just artificial categorization based on how we want to organize those 10 steps, making it as simplified as possible instead of running through all 10 steps. I personally love to look at it as just 10 steps, but when things get too complex, we simplify it a little bit and break it down into smaller pieces. So those full 10 steps are broken down into smaller pieces in two phases known as the preparatory phase and the payoff phase. We have established that there are two phases in which glycolysis is considered, which is the preparatory phase and the payoff phase. Right here, we want to look at the full glycolysis overview and be able to distinguish which part of these 10 steps of 10 reactions in glycolysis are for preparatory phase, which is the first 5 steps, and which of the steps are for payoff phase, which is the later 5 steps. Now, on this diagram, you could see the glycolysis overview is starting with the substrate glucose. So glucose undergoes a 10-step wise reactions showing from the glucose to pyruvate. Now, in glucose is converted to glucose 6 phosphate, and from glucose 6 phosphate to fructose 6 phosphate, and to fructose 1,6 bisphosphate. Now, in fructose 1,6 bisphosphate, you have a split giving you glyceraldehyde 3 phosphate and dihydroxyacetone phosphate, which is designated as GAP and DHAP. Now, this step, glucose glucose 6 phosphate, fructose 6 phosphate, fructose 1,6 bisphosphate, splitting right down the middle into 3 carbon 3 carbon as glyceraldehyde 3 phosphate and dihydroxyacetone 3 phosphate is what forms the first 5 steps, known as what the preparatory phase. Then this glyceraldehyde 3 phosphate goes down the reaction to pyruvate, known as giving the second 5 steps. Now, this is the general overview. So, in the next slide, we'll be looking at the, the steps in the glycolytic pathway. So, glucose conversion of glucose to glucose 6 phosphate is step 1. Glucose 6 phosphate to fructose 6 phosphate, step 2. Fructose 6 phosphate to fructose 1,6 bisphosphate, step 3. And the splitting of fructose 1,6 bisphosphate into 3 carbon each right down the middle gives step 4. Step 4. Now, glucose dihydroxyacetone is an isomer of glyceraldehyde 3 phosphate, and that is broken down, is converted to glyceraldehyde 3 phosphate through an isomerase enzyme, and that gives another molecule of glyceraldehyde 3 phosphate. So that first 5 steps gives you what? The preparatory phase. Now, glyceraldehyde 3 phosphate moves from is converted to 1,3 bisphosphate glycerate, and that is further converted to 3-phosphoglycerate, 2-phosphoglycerate, and phosphoenol pyruvate. Phosphoenol pyruvate is finally converted to pyruvate, and that's from glyceraldehyde 3-phosphate down to pyruvate, gives the second phase of the glycolytic pathway, known as what, the payoff phase. So this slide just shows you clearly the 10 steps, how in 10 reactive steps in glycolysis. There are specific reactions which shows how the ATP is formed. um I've previously established that 2 ATP is invested and 4 ATP is produced. Let's see how that is done. In this slide, you will see also the 10 steps that we have established, but you begin to see where the ATP is invested. In step 1, you see that glucose converting to glucose 6 phosphate uses one molecule of adenosine triphosphate, which is ATP, and gives its inorganic phosphate to glucose, thereby forming glucose 6 phosphate. So the inorganic phosphate given off at this point is attached to the carbon 6 of the glucose molecule, and that gives you what? Glucose 6 phosphate. So ATP is used up producing ADP. That is for the step 1. Look at step number 3 again, the conversion of fructose 6 phosphate to fructose 6 bisphosphate. That is also another point where another molecule of ATP is being used up, and that one molecule gives the inorganic phosphate to fructose fructose 1 phosphate. So you have fructose 1,6 bisphosphate or diphosphate, showing that there are now two phosphate group in the fructose 1,6 bisphosphate or diphosphate. Of course, glucose is converted to um, fructose 6 phosphate by the enzyme isomerase. So you um you have that produced there as fructose 1,6 bisphosphate, and this fructose 1,6 bisphosphate is split right down the middle to form your dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, giving three triose triose sugar from fructose 1,6 bisphosphate. Of course, the glyceraldehyde 3-phosphate now moved down to um pyruvate in another 5 steps. Now we look at the specific enzymes that catalyzes this reaction, and um in the specific enzymes we look at glucose. Starting from step 1 down to step 10, you begin to see each one of these enzymes. Your first reaction in the first step is catalyzed by exokinase. The exokinase is a kinase that uses the inorganic phosphate that is given up from adenosine tri- triphosphate, and it attaches that inorganic phosphate to glucose, giving glucose 6 phosphate. So ATP is used up at this point. That enzyme is hexokinase. In your step 2, you have the enzyme uh, phosphohexose isomerase. I earlier told you that is an isomerase enzyme that converted glucose 6 phosphate to fructose 6 phosphate. So glucose 6 phosphate and fructose 6 phosphate are isomers. Isomerase enzyme converts this glucose to fructose. The third enzyme in which another ATP is invested, or in the preparatory phase, is the fructose 6 phosphate to fructose 1,6 bisphosphate. Fructose 1,6 bisphosphate here is achieved by the kinase fructose phosphofructokinase 1. Phosphofructose kinase 1 is another kinase enzyme, just as hexokinase in step 1. In step 3, another kinase comes in here and uses the inorganic phosphate produced from the conversion of ATP to ADP, and that inorganic phosphate at this time is attached at fructose carbon 1 of the fructose molecule, thereby giving you fructose 1,6 bisphosphate. And the fourth enzyme is the aldolase enzyme, and the aldolase enzyme is the one responsible for the splitting of the fructose 1,6 bisphosphate right down the middle, producing 3 carbon units each of the triose sugar glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Now, the dihydroxyacetone phosphate is also an isomer of what? The glyceraldehyde 3-phosphate, and that shows in step 5 by the enzyme triose phosphate isomerase. The isomerase enzyme converts the dihydroxyacetone phosphate to glyceraldehyde 3-phosphate, and that shows the first 5 steps, which is the preparatory stage phase or the investment phase of the glycolytic pathway. Now, from step 6, you begin to look at the payoff phase, whereby ATP is being produced. Remember or recall that in step 1 to 5, 2 molecules of ATP was invested or used up in the process, and in this step 6 to 10, we have ATP being produced. Now, let's look closely at step 6. Step 6 is being catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase, which uses the coenzyme in its oxidized form NAD+. NAD+ is reduced to NADH, and PI is being inorganic phosphate is being introduced at this point, and that's for step 6, and that produces leads to the conversion of glyceraldehyde 3-phosphate to glyceraldehyde 1,3-bisphosphate, while the inorganic phosphate being produced is attached to the carbon 1 of this triose sugar, making it 1,3-bisphosphate. Now, at this point, 1,3-bisphosphate is converted to 3-phosphoglycerate. 3-phosphoglycerate. Now, this ADP is converted to ATP. Now, the first molecule of ATP is produced at this point, and you have 3-phosphoglycerate. That reaction is catalyzed by the enzyme phosphoglycerate kinase. That's step 7. In step 8, is catalyzed by phosphoglycerate mutase, where 3-phosphoglycerate is converted to 2-phosphoglycerate. This enzyme is just responsible for the shift in the phosphate group from position carbon number 3 to carbon number 2. A mutase enzyme is responsible for that. That's why you have the enzyme as what? Phosphoglycerate mutase. Now, phosphoglycerate mutase at this point has gives off one water molecule to form phosphoenol pyruvate, and that enzyme is being catalyzed by enolase, the enzyme enolase. So at this point, you have a dehydration reaction where water is being given off to produce what? Uh, Phosphoenol pyruvate. Now, phosphoenol pyruvate is now catalyzed or converted to pyruvate in the 10th step of the glycolytic pathway, and that enzyme that catalyzes that step is also another kinase enzyme, pyruvate kinase, whereby ADP is being used up to produce what? ATP. So you see this glyceraldehyde 3-phosphate going down to from step 6 to step 10 is produced what? 2 molecules of what? ATP. 2 ADP is used up in this process, and 2 ADP is what? Produced. Now, this dihydroxyacetone phosphate in step 5 also produces here another glyceraldehyde 3-phosphate that also goes through same process. So this process and this process are exactly the same. So when it goes through this same process, it produces another two molecules of what? ATP. And that makes the payoff phase to have what? Four molecules of what? ATP being produced, producing for a total of four molecules of energy of ATP. And the net ATP being produced in all is now two molecules. Why? Because the payoff phase produces four, and remember, in the investment or preparatory phase, two ATP were used up. So if you minus these two ATP from the four produced, you have a net production of ATP from the 10 steps of glycolytic pathway as what? Two ATP being produced. In our next set of lectures, we'll begin to look at the exact structure of the 10 steps in glycolytic pathway. Thank you.